Hi, thank you for joining us. My name is Deborah Honig, and I'm a partner at Adelaide Capital, an IR firm focused on small cap Canadian value and growth companies. Today I'm speaking with Rahim, portfolio manager at Next Edge Capital. For those of you who don't know Eden, he's one of the most renowned institutional investors in the biotech sector in Canada. And Eden is going to answer some of my basic questions about biotech investing. Make sure you pay attention to the end when I ask the biggest question of all. So Eden, thank you very much for joining us today.
Deborah, thank you for having me. I'm delighted to be here.
As you know, I'm a generalist, so I happen to know very little about most things. And I'm a salesperson too, so I can usually only remember 2 or 3 things. But you and I both agree that it looks like we're heading into a bull market for biotech investing. I thought it would be worthwhile to have you answer some of my basic questions that I'm sure a lot of investors out there would either like a refresher on or have no idea what the terminology is. So thanks. I'll start off with the first question I have is, what is the typical preclinical process?
If you look at a 10-year process to discover a drug to commercialization, there are many phases. The earliest phase is when you have a hypothesis as to what might work in targeting a certain disease or certain indication. And so you experiment with a variety of molecules or a variety of proteins to see if that would work to potentially, inhibit or trigger the production of a certain protein. But first, before you can begin testing in humans, you must test it in animals for 2 things, because you want to understand, one, is that hypothesis nearly correct? Does this protein actually impact the production or the suppression of a certain protein? And hopefully it's not killing the mice or primates or anything like that. So once they do that, they begin testing in animals, and you have specific defined models. So if you test mice following a certain model, you test primates following a certain model, and from that you gather information such as what might be acceptable dosing, what the mechanism of action of that drug is, and you gather this information and you use that to file something called an IND, an investigational new drug. And once you file that with the FDA, that enables you to begin testing in human beings.
And that would be clinical trials.
That's correct.
Are most trials and primate trials indicative of what results for human trials might look like?
To a small degree. It's very different, and how a drug acts when it gets into a human is very different. Really what you want to see is you want to determine whether the hypothesis as to how it works, or whether targeting this receptor generates some activity, is predictive of how it might be, is what you're looking for. Now, there are a lot of small companies that make very grandiose claims of preclinical models and phase 1 models. Part of my investing process is to ignore that completely. It's merely indicative. When it gets into human beings, it's a whole different ball of wax.
Okay, and what is the difference between phase 1, phase 2, and phase 3 trials?
Sure. So, as I mentioned earlier, the process of discovering a molecule or a biologic and getting to commercialization is roughly 10 years. So once you've entered the clinic for clinical trials, you begin a phase 1 trial. That's the FDA allows you to test the drug in humans. But all you're testing is to make sure it's not toxic. So you're measuring toxicity and certain side effects and so on. And you're testing various doses. You want to see, does a small dose, a medium dose, or a high dose have different effects? Is there a dose ranging effect? And is it even effective at all? So that's what you're trying to measure from it. Phase 2 is when you escalate up and you're trying to determine something called efficacy. And phase 2 is broken up into two areas, phase 2A and phase 2B. Phase 2A is open label, which means the investigator and even the patient may know that they're getting the experimental drug. Phase 2B is much more important because that's determining whether or not the drug against a standard of care is effective. And that's, for me, that's the most important trial in determining whether someone like me invests in the company in the future. Because if you do a really good phase 2B, double-blind placebo control, neither the investigator nor the patient knows whether they're getting the experimental drug or the standard of care, called placebo. If it's efficacious, then you know you legitimately have a real drug for the very first time, right? Because now it's against standard of care and it's blinded. But the failure rate is highest in phase 2B. The failure rate's about 70%. That's why, that's exactly why it's the defining point. I typically invest after phase 2B results, because then you know you have a drug that may potentially have some commercial success. Phase 3 is when you take the information from your phase 2 success and you designed a trial for phase 3. This is what you're going to submit for FDA approval. And that typically runs 2 or 3 years in duration, and it's the highest cost because you're dealing with a large number of patients, highly specific patients that have been selected based on the information from your phase 2. And the outcome of that phase 3 trial is what you will submit to the FDA. And for most standard indications, you require 2 large phase 3 trials. For rare diseases and so on and other indications, which we talk about later, you just need 1. But for the most part, 2 large phase 3 trials, and that will run another 2 or 3 years.
So what is the whole process? Like, how long does that take typically?
That will take, so a preclinical will take maybe about a year. Phase 1 will take about another year. Phase 2 will typically take about 2 years or more, 2 to 3 years. And a phase 3 will typically take 2 to 3 years. And then you have the submission process, which takes another 6 months or so before you get to the point where the FDA can review your package.
Wow, it's like building a mine.
It is. But this will factor in to why M&A is such an important component of biotechnology, because of this costly, lengthy period of development that's required. Then at the end, you may fail.
So when you were talking about the different phases, you mentioned a term called standard of care. What is a standard of care?
So for any particular indication, whether it's let's say it's breast cancer or it's Alzheimer's, there are drugs that are approved that are used by physicians and key opinion leaders to treat these patients. So that's your benchmark, right? So the drug, assuming that you're going for superiority, has to perform better and safer than the drugs that are currently in use for that particular indication. So that's a standard of care.
Okay, and another term that I hear quite often is orphan drug status. Can you explain what this is?
So there are a variety of different designations that the FDA assigns to what you file and what approval that you get. So orphan drug designation is when a company has a drug that they think treats a small patient population, which is for orphan drugs defined by less than 200,000 potential patients. They submit, I think it's 7 to 10 question meeting certain requirements. They submit it to the FDA saying we request orphan drug status because it's a small patient population. And so they apply for it. It's not an approval or disapproval type thing in terms of clinical trials, but the FDA issue these fairly easily, right? You have a drug, small indication you're targeting, with the expectation that later on you'll target a larger indication. So, it's important, but is not as significant as a lot of small companies play it up to be. Because again, it's a simple questionnaire. Once you've met the requirements, the FDA will issue orphan drug status, which gives you some exclusivity It gives you some favorable tax rebates because the FDA wants to incentivize companies to develop drugs for small indications that don't have a high commercial value.
Okay, that makes sense. And what is fast track status and how does the company get it?
So fast track status is when the FDA deems that the therapy that you're developing in clinical trials addresses an unmet need. It addresses a group of patients that may be at higher risk. There is the treatments that are available are highly deficient compared to what the potential of this drug may be. So the FDA, instead of the usual sort of 10-month period of review, will allow this drug to be reviewed for a 6-month period. So once you submit your filing for approval, the FDA will review it within the 6-month period versus the usual 10 months.
So you hear a lot about emergency use.
Yes.
And what's the other term that they kick around? Compassionate care, something like that?
Yes.
Explain this and exactly how it works.
Sure. So compassionate care applies to a drug that is approved for another indication. But because of emergency situations, they decide that even though it's not approved for the indication that you want to use it in this case, treating patients that have been infected by the coronavirus, they will take a drug that's approved for something else and apply for approval to use it in this particular indication, even though there have been no clinical trials to prove its efficacy and so on. With EUA or emergency use authorization from the FDA, you've seen a proliferation of companies that have designed diagnostic tests, right? Now, the efficacy and the specificity of these tests have not been clinically proven. But nevertheless, the FDA has allowed these tests to get to market because the need is so urgent to have anything that could potentially spot or detect the coronavirus in infections. The problem with that is that it's essentially marketing without clinical approval. So the FDA allows you to go and proceed, and you can make, again, claims that are not necessarily backed by a body of clinical work. So what's really important, that creates a huge need for companies or a company that can validate those tests, determine false positives from false negatives and so on. And there's a need for that out there because otherwise there is no other way to justify the claims that some of these diagnostic test companies are making.
Those are considered medical devices, not drugs.
That's correct. It's a different approval process for that.
And now just switching gears, so what is immunology?
So traditionally, immunology has been the study of the body's ability to develop a defense mechanism against viral infections and so on. That's traditionally how it's been. But over the last decade, we've seen immunology actually evolve to include oncology. So we've had sort of immuno-oncologic drugs developed whereby your body's immune system, which traditionally it has not been able to do, your body's immune system can be enlisted to target cancer, which traditionally you relied on chemotherapeutic drugs and so on. So now you can trigger your body by decloaking cancer cells, and your body can now go by and recognize that this as a cancer cell and attack it. And so that's been the biggest breakthrough, and that's been the biggest advance in immunology over the past decade.
And what is RNA?
So RNA, so backtracking a step, we all heard about DNA, right? So you think about a ladder with 3 billion steps on it, right? And so DNA is double-stranded, and each of those steps are base pairs. You've got an alphabet, which is basically the software, our human software. So you've got A that binds to T and C that binds to G. And so think about that 3 billion step ladder. What RNA is when you want to convert that DNA into a protein, you have to go through a process of replication and translation. So the latter breaks into half, and then it pairs up with the complementary RNA. So RNA forms by binding to each of those single strands of DNA that's been broken up. That creates an RNA strand. And that's what's used. That single-stranded RNA is what's used to create proteins in our body. The reason why this is topical now, it's because the coronavirus is an RNA strand. And the RNA strand that it uses your body's ability to convert RNA into proteins. It exploits that. And so the coronavirus is a single RNA strand, which is why it's so topical right now. So you've got drugs like Gilead's remdesivir. They target the replication of single-stranded RNA, right? And so it targets that. And so ordinarily, RNA is a good thing. But in the case of the coronavirus, because it is actually a single-stranded RNA, inhibiting that is a good thing.
I think I understand most of that.
Yeah.
But now we get to move on to the section that I'll understand way more about.
Sure.
Investing.
Yeah.
So what makes a good biotech investment, in your opinion?
So that is, again, eye of the beholder. There are different managers and different investors that like different components. Some are very early stage. Some like to invest in early stage phase 1 companies, some preclinical. For me, based on, again, maybe it's a bias from my option background, I think a good biotech investment where you don't have to rely on management's projection of how effective a drug is, but you can rely on the actual clinical data. So I like to wait until after phase 2B data is available. And so the company is substantially de-risked, and you can invest, you can earn hundreds of percent as it appreciates through the phase, from the post-phase 2 through the phase 3 into approval process. And so for me, I look at it as a risk-reward basis, and the fact that I don't have to rely on management's hyperbole as to how good a drug is, but I can rely on the actual clinical data, and then I can speak to key opinion leaders, KOLs, as to what their view of what that clinical data is like. And so that's my particular niche in investing and what I think makes a good biotech investment.
So which areas of biotech do you see the biggest opportunities right now, and which areas would you be the most cautious about?
Well, I think that changes over time as we see breakthroughs develop in biotech. So 20 years ago, I was a big early investor in HIV therapy breakthroughs and in oncology. And I was an early investor in companies like Gilead, IDEC Pharmaceutical, all that developed huge blockbuster drugs in HIV and in oncology. I am much less so now. I don't have any HIV investments because, again, we have saved tens of thousands and perhaps hundreds of thousands of lives by the drugs that biotech has developed in HIV, and it's now down to a vaccine, Truvada from Gilead, which is amazing. And oncology, similarly, we've seen these enormous breakthroughs in cancer therapies. And so it's very, very crowded in oncology. There are thousands of trials. There are hundreds of companies working on it. The bar is very high. The breakthroughs have been substantial. So the failure rate for me is much higher. The areas of focus for me are in CNS, central nervous system indications, Alzheimer's disease, Parkinson's disease, schizophrenia, epilepsy, bipolar disorder. I think that's where the needs are greatest. That's where the bar is potentially lower for therapies to become successful. And so that's where I'm focused on. Again, it's not that I'm against investing in other areas. I just think that the breakthroughs have been so substantial in those other areas that I've now moved on to where I think the large unmet needs are.
Well, I'm sure everyone wants to know about what your COVID investing strategies are, but I'm not going to let you tell them until my conference.
Okay.
But I guess the biggest question that I have, and I think a lot of investors have that don't invest in this space, is like, don't most biotechs fail? And is it really worth the risk?
Absolutely, and that goes back to the optionality we talked about early on in the conversation. Absolutely, most biotechs fail. The success rate from phase 1 to commercialization is 10%. It depends on the phase. If you hold directly from that phase, that's what your success rate is. And there are lots of hurdles and lots of financings along the way that you have to endure. You can break it up into phases, and you can be an expert phase 1 investor and not invest in later phases. You're not taking, if you're an early stage investor, you're not taking a lot of the onerous clinical risk of phase 2 and so on. And I know lots of investors that focus on that earlier stage and they do well. As for me, I focus on late stage because I think the risk reward is good. I know you have legitimately a drug that can become a commercial success, as well as have a significant improvement on the quality and the survival of many lives. And I can sit on a foundation of clinical data for that. So for me, if you go to phase 3, the success rate in phase 3 data is 60%, right? So I'm comfortable with that. And you can increase that even more by following a series of other disciplines that we follow. So then your success rate goes to 70% or 75 or 80%. So yes, the failure rate's high, but it depends on where you sequester your investing focus into. Then you can make the success rate very high versus a high failure rate.
Well, that makes a lot of sense to me, Eden. Thank you. Thanks for taking the time today. For those interested in hearing more about Eden's fund at Next Edge Capital, I encourage you to visit their website, nextedgecapital.com. And as I mentioned earlier, Eden has kindly offered to do the keynote speech at Adelaide Capital's conference, COVID and Healthcare. It's a virtual conference, so check out our website if you want details. And his speech is going to be called Signal from the Noise, a Compass to Finding COVID-19 Investment Opportunities. And thanks again for your time today, Eden. I found it really informative.
It was terrific, Deborah. Delighted to be here.